The overall goal of this combination of standard methods is to demonstrate how the C. elegans intestine can be used for the in vivo analysis of polarized membrane biogenesis and lumen morphogenesis at the single-cell and subcellular level.
This method can help answer key questions in the polarity and tubulogenesis fields, such as how apical versus basolateral membrane domains and other subcellular asymmetries are established and maintained during and after morphogenesis.
The main advantage of this model system is that this transparent single-layered epithelium of only 20 cells can serve as an in vivo tissue chamber to dissect single-cell polarized membrane and lumen biogenesis.
The techniques can be extended to the analysis of other C. elegans morphogenesis phenotypes. The focus here is on the analysis of membrane biogenesis, specifically on apical membrane and lumen biogenesis.
The procedures include refined imaging techniques in fixed and live animals to distinguish membrane domains and subcellular components, and to identify lumen morphogenesis phenotypes in the C. elegans intestine.
This video on intestinal tubulogenesis is accompanied by a video on excretory canal tubulogenesis, mutually informative models for the in vivo analysis of polarized membrane biogenesis. With regard to labeling, the generation of transgenic animals with fluorescently labeled fusion proteins for live imaging is described in the accompanying video on the analysis of excretory canal tubulogenesis. Such animals can be directly used in RNAi experiments. Their generation